In this presentation, we'll take a look at the cells that make up the nervous system and how they communicate with one another. Nervous systems, whether they're in higher organisms like humans or lower organisms like insects, for example, all share the same function. Their function is to process information, and they do that in three stages. There's first the sensory input, so the organism senses something in its environment. There's integration, which takes place either at the level of the brain or the ganglia, or collections of nerve cells. And then there's some kind of a motor output. The name that we give to the cells that conduct information within the nervous system is the neuron. And neurons can use either electrical signals or chemical signals to communicate with one another. And we'll look at how both of those work. In this schematic, you can see how the nervous system is described in higher animals. We have the central nervous system, which takes place in the brain, the central nervous system, which includes the brain and the spinal cord, and then the peripheral nervous system, which includes all of the nerves that branch from the spinal cord. So, for example, you might have sensory information right now coming in from your fingertips, telling you that you're holding a pen. That sensory information comes from peripheral nerves that stretch to your fingers back to your nerve cord. Your nerve cord would send that information for integration to your brain. Integration is that process whereby you assign meaning to the sensory input coming from your hand. So you determine that holding that pen is what your neurons are detecting. You assign meaning to the feeling that you have in your fingertips. So that's integration. That takes place in your brain. And that might cause you to do some kind of motor output, like you might start writing with your pen. So that information comes from your brain, travels to your spinal cord, and then back down to your fingers. So this, of course, is a very generalized schematic, but it helps you to understand where nerves travel and where nerves carry their information. From the outside world to your central nervous system, to your brain, and then back out to you so that you can respond to your environment. We assign names to neurons based on their role in that scheme. So the sensory neurons are those that detect stimuli from the environment. The interneurons are those that help you to integrate that information, so those tend to be located either in the brain or in the ganglia. And then there are the motor neurons, which trigger your muscles or your glands to respond to that external stimulus. In addition to neurons, there are cells located in the nervous system that support the neurons. These fall into a few different categories, but we call them all glial cells. We'll start with the ependymal cells. The ependymal cells line the ventricles that surround your nervous system organs, so the brain and the spinal cord. And those cells enclose a fluid called the cerebrospinal fluid, CSF for short. The cerebrospinal fluid provides a lot of cushioning for the organs. Obviously, your brain and spinal cord are very important organs in your body, and so the CSF provides some cushioning and protection for those organs. So the ependymal cells are really there to enclose the CSF and to help it to circulate. There are microglial cells, which protect your nervous system from microorganisms or parasites that might infect it. There are two kinds of cells that myelinate the axons on your nerve cells, and those are called oligodendrocytes and Schwann cells. A little bit more on those later. Astrocytes are cells that are shaped like stars when you look at them under the microscope, hence their name. They maintain a structure called the blood-brain barrier. The blood-brain barrier is a collection of blood vessels that essentially filter any blood coming to your brain. If you think about how important the brain is, how pivotal the brain is to animal functioning, it becomes obvious that your body would want to protect it. And so the animal's solution to this problem is the blood-brain barrier. This collection of blood vessels provides protection for any blood that's going to circulate to your brain. So if there is some sort of an invader in your blood, any kind of a pathogen, the blood-brain barrier attempts to catch it. Now there are some places where the blood-brain barrier does well, and there are some where it doesn't succeed very well. For example, Alcohol is a toxin to your brain, and yet the blood-brain barrier does not prevent alcohol from traveling straight to your brain, and that's why alcohol can do so much damage. Finally, last type of glial cell we'll look at are called the radial glia, and these are mostly relevant in the synthesis or the development of the central nervous system. It's a little more detail than we'll get into. We won't talk specifically about how the central nervous system is created beyond what you already know, that it arises from the ectoderm of the developing embryo. Now that you know about the different kinds of cells that support neurons, let's take a closer look at neurons themselves. Their structure is quite unique among animal cells. We'll start with the cell body. It's also called the soma. You'll see it referred to in both ways. The cell body is really what we recognize as being the cellular part of a neuron. It's what we most recognize as being a cell. So it's surrounded by a cell membrane, it has the nucleus, and it has the organelles. And this is where most of the cytoplasm is located, in the cell body. What really makes a neuron unique, though, are the projections arising from that cell body. And they come in two different varieties. There are dendrites, which tend to be short and branching, and one neuron can have many, many dendrites. And then there are axons, which tend to be longer than dendrites, and although neurons can have more than one axon, they tend to have fewer axons than dendrites. The dendrites and the axons have both function in communication, but they have opposite functions. The dendrites are the receivers of information for the neuron, whereas the axons are the transmitters of information. So whereas an axon would send a signal, dendrites would receive the signal. So here we have a diagram that shows two neurons connecting to each other. In here we have the axon hillock giving rise to the axon, and this neuron here appears to be sending a signal to this neuron because its axon is projected onto its dendrites. That makes this the presynaptic neuron, and it makes the second neuron, the receiver, the postsynaptic neuron. Between them sits a tiny bit of empty space right here. The region where one neuron is in contact with another is called the synapse. And let me explain contact. Contact does not mean touching in this case. Neurons do not generally touch one another. In fact, there's a tiny bit of empty space in between the two neurons. So here we have the axon of the presynaptic neuron and the dendrite of the postsynaptic neuron. You'll notice there's some empty space in there. That's called the synaptic cleft. And if one neuron wants to communicate with another, they don't close the cleft. The cleft is always there. But instead, the presynaptic neuron will release tiny chemicals called neurotransmitters onto the dendrites of the postsynaptic neuron. And that's how one neuron will communicate with another. You'll recall that we talk about cells as having a natural resting membrane potential. This is a review of the cell membrane and cell structure. The inside of a cell tends to be about negative 70 millivolts in comparison to the outside. So it has a slightly negative charge on the inside in comparison to the outside. And that's simply because the outside tends to have more positive ions than negative, and the inside tends to have more negative ions than positive. This is especially important in neurons because neurons need to communicate using electrical signals. And so it's very vitally important that the resting potential in a membrane of a neuron be maintained. Remember that ions face an electrical gradient, whereby positive ions are attracted to a negative side of the membrane, but they don't always diffuse across the membrane in that direction. And so in order to maintain this differential, this charge differential, this area of positive and this area of negative charge, we have to have some kind of pump. And the solution to this problem is the sodium-potassium pump. This is a review, but let's take a look at what the resting potential of a cell really looks like. On the bottom here we have the inside of the cell. And you can see that we have many potassium ions located on the inside. We have a few sodiums, but by far the potassium really outweighs here. So we got lots of potassium ions on the inside, very few sodium. On the outside of the cell, we have the opposite scenario. We have lots of sodium with very little potassium. So think about the chemical gradient that we've set up here. Lots of potassium ions on the inside. Well, chemically, where do they want to diffuse? They want to diffuse outside of the cell. And there are certain ion channels that are always open, and they simply allow the diffusion of potassium ions outside the cell. And oppositely, the diffusion of sodium occurs through sodium channels that are always open in the cell. So how do we manage to maintain this resting potential, this slightly negative charge on the inside, when we have all of this unregulated diffusion taking place through these open ion channels? Solution is the sodium-potassium pump. The sodium-potassium pump makes sure that, on average, there are more sodium ions on the outside, even though they manage to diffuse across the membrane. How do they do that? Well, they use active transport with ATP, so it expends energy, to pump 3 sodium ions outside the cell, and at the same time they pump 2 potassium ions back into the cell. So even in the face of this electrochemical gradient that forces potassium outside the cell and sodium in, the sodium ion pump manages to make sure that sodium net stays on the outside and potassium net stays on the inside. And it does so using ATP. A close look at some videos that explain this resting potential and how the sodium-potassium pump really helps to maintain it.
Unstimulated, neurons maintain a constant electrical difference, or potential, across their cell membranes. This potential, called resting potential, is always negative inside the cell and ranges from -40 to -90 millivolts. If a neuron is stimulated, the negative potential inside the neuron can be made either more or less negative depending on the stimulus. If potential is made sufficiently less negative, it reaches a level called threshold, and an action potential is triggered. During the action potential, the neuron suddenly becomes 20 to 50 millivolts positive inside. Action potentials last a few milliseconds before the cell restores its negative resting potential. The cell membrane of a neuron encloses cytoplasm with various ions dissolved in it. The neuron itself is immersed in a salt solution, the extracellular fluid. The ions of the cytoplasm consist mainly of positively charged potassium ions and large negatively charged organic molecules, such as proteins. Outside the cell, the extracellular fluid contains mostly positively charged sodium ions and negatively charged chloride ions. Since charged particles cannot pass through the lipids that make up cell membranes, they must travel through channel proteins that extend through the membrane. In an unstimulated neuron, only potassium ions can cross the membrane, traveling through specific proteins called potassium channels. Although sodium channels are also present, in unstimulated neurons, they remain closed. Since only potassium ions can cross the membrane, and potassium ions are more concentrated inside the cell, they diffuse out of the cell, leaving the large negatively charged organic ions behind. As more and more positively charged potassium ions leave, the inside of the cell becomes increasingly negative. But since opposite charges attract, as potassium ions diffuse out, an electrical force develops that tends to pull them back inside. At some point, the diffusion of potassium ions out of the neuron due to concentration differences is balanced by the electrical attraction tending to pull them back inside. This is the point at which neurons reach resting potential. Reaching resting potential in this way does not require significant changes in the potassium concentration inside or outside the cell. Only about 1/10,000th of the potassium ions initially inside a neuron must leave to create a resting potential of -60 millivolts. But it is action potentials, not resting potentials, that carry information through a nervous system.
So that's sort of an overview of why the membrane potential is so important when it's at rest, because the neuron really needs to be able to transmit the change that comes with an action potential. And let's take a look now at the sodium-potassium pump that enables the maintaining of that resting potential. So here we have the sodium-potassium pump. Notice 3 sodium ions need to bind. Nothing's going to happen, though, until ATP comes, phosphorylates the protein, and forces it to change shape. Once that occurs, the sodium ions are released to the outside. And now we'll wait for 2 potassium ions to come and bind. Now we need to dephosphorylate so that it can revert back to its original conformation. And the potassium ions will be released into the inner cell space. So again, this is a form of active transport because it requires the use of ATP as an energy source. And you can see that the reason ATP is needed is because we need a conformational change in the shape of the membrane protein that comes with phosphorylation. We've referred a couple of times now to this action potential. Let's take a close look at what it really means. Strictly speaking, an action potential is a rapid change in the polarization across the membrane of a neuron. I'll say that again. An action potential is a rapid change in the polarization across the membrane of a neuron. So how do we get that rapid change? It can only result from the movement of charged ions across the membrane. And remember that that movement is really happening all the time. We always have some sodium diffusing in, some potassium diffusing out, and we always have the sodium-potassium pumps pumping the ions in the other directions. So what's the purpose of this action potential? Well, this rapid change in the polarization of the membrane turns out to be how neurons communicate with one another. If I want to cause a change inside of a neuron, the fastest way to do it is using an electrical change, by changing its polarization. We've talked a lot about how cells communicate. Some cells communicate with one another by causing a change in gene transcription. But that takes an awfully long time to have any kind of an appreciable effect on the organism. But a change in the current, or a change in the electric potential across a membrane, takes just milliseconds to occur. Okay. You'll notice the time scale for this action potential is on the order of a few milliseconds. And it's so vital because as animals, we need to respond to our environment so rapidly that an action potential has turned out to be the most effective, most efficient way that our cells have of communicating with one another, at least in the instance of reactions that need to happen instantaneously. So the action potential is a signal that an axon carries along its length and sends to its next cell. So we'll take a look at how that happens exactly. We'll start with a cell that's already at its resting potential. So some of the potassium channels are open, and we can see potassium diffusing. They're not shown, but normally if a potassium ion channel is open, and so we have some potassium ions diffusing across the membrane. But for the most part, the gated ion channels are closed, and they're gated because they're at resting potential. So it's the resting potential of the membrane that keeps those particular ion channels closed. Meanwhile, we are maintaining a negative charge overall inside the cell and a positive charge overall outside the cell. So if we correlate this to our membrane potential graph here, at resting potential you can see we're sitting a little bit below negative 50, probably around negative 70 millivolts or so at resting potential. So this is before any kind of an action potential has arrived or occurred inside the cell. The next stage is called depolarization, and you can see that it's one of the longer stages of an action potential. It takes a while to get going. In response to a stimulus, probably from another neuron, the sodium channels along an axon will open. And think about where sodium wants to diffuse. Sodium is super concentrated outside the cell. Furthermore, it's facing a positive charge outside the cell, and sodium is repelled by those positive charges. It really is seeking a negative charge. So in response to the opening of sodium channels, sodium diffuses into the cell, and that's why we see this rise in the membrane potential, because sodium is a positive charge, and so as it comes into the cell, we see an increase in the charge inside the cell. So it was at negative 70. By adding some positive charges, we're raising it to about negative 50 or so. This part of an action potential is vitally important because if just a little bit of sodium diffuses in, if those sodium channels maybe close after a couple of seconds, we won't reach the critical value called the threshold value for an action potential. If we fall short of the threshold value, shown here at about negative 50 millivolts, if we fall short of that, then no action potential will occur. What will happen instead is the sodium ion channels will simply close and no action potential will take place. But if enough sodium ions come into the cell and enter the cytoplasm, and we reach that threshold value, then an action potential is guaranteed to take place. This threshold value is why we call an action potential an all or none phenomenon. It either takes place or it doesn't. You don't have some big action potentials and some small action potentials. It either takes place or it does not. So this depolarization stage here is vital. Assuming that we do reach the threshold value, we get this rapid influx of sodium ions because in response to that slight change that we saw during depolarization, even more sodium ion channels open. It's a positive feedback loop. In response to some sodium ions coming in, more sodium ions will be able to come in because more gates open. So because of this positive feedback loop, we get increasing numbers of sodium ions rushing into the cell, and we get this rapid changing, this rapid rise in the membrane potential. And this is the actual action potential happening right here, this maximum value for the charge inside the cell. In fact, it gets so large because so many sodium ions rush in that the charge actually becomes positive inside the cell relative to the outside. So you can see we not only hit our threshold value and raised our internal charge up to 0, we actually passed 0 and are now in a positive range because so many sodium ions have rushed inside the cell. So this action potential is happening. Once the action potential is carried, the cell needs to return to its normal resting potential. And so we basically see the opposite thing happening. Whereas before, the sodium channels opened, once the action potential value is reached, here it appears to be about positive 30 millivolts or so. Once that's reached, immediately the sodium channels become inactivated and they close. It's a very fast process for sodium channels to close. And so you see them here in light purple. The sodium ions, in response to encountering this positive charge on the inside, close. Meanwhile, the potassium channels are still open. And so the potassium ions, which are not so happy inside the cell because, well, gosh, we got this positive charge here, and now there are so many potassium ions on the inside. The potassium ions move outside the cell. And this is why we start to see a drop in the membrane potential, because we're losing the positive potassium ions that are inside the cell. So they rush out very, very quickly. As a result, the inside becomes negative again. So we call this the falling phase of the action potential, where the positive charges are leaving. This happens so fast because the cell at resting potential is more permeable to potassium than it is to sodium, just because there are more open potassium channels. And so what happens is a little bit more potassium than we had hoped for actually escapes from the cell. So that's called the undershoot here. But slowly, as more potassium gates close, the membrane potential eventually returns to its resting state. And so this period here is called the refractory period, when no new action potential can be generated inside the neuron, simply because we're still recovering, we're still closing some of those potassium channels, and the ions are still diffusing down their electrochemical gradients. So during that refractory period, no more action potentials can act on the neuron. So this prevents a neuron from, say, having two action potentials simultaneously. That action potential can occur inside an axon, but it needs to travel down the axon. Remember that axons are incredibly long, especially when you compare them to the size of the soma, or the cell body of a neuron. Very great distance for an action potential to travel. And so it really needs some kind of insulation along the axon to ensure that the electrical gradient isn't lost. Think, for example, about the power cords for your MacBook. The power cord is insulated by something that does not conduct electricity at all. It's insulated by a plastic layer. Of course, electrical current needs to travel inside the cord, but on the outside it's insulated by something that doesn't carry that charge at all. That's so that it can trap the electrical current inside the wires, inside the metal conducting wires. Similarly, we insulate an axon so that it can more efficiently transport that action potential down the axon, so that no charge is lost along the membrane. The insulating layer that we put along the axon is called the myelin sheath. And it's made by oligodendrocytes if it's a neuron in the central nervous system, or Schwann cells if it's along axons that are located in the peripheral nervous system. So if you zoom in here and you take a look at the structure of the axon, you see that the myelin comes in these kind of bulk, these kind of sheaths here. And there are actually breaks in between the myelin sheaths. Those are called the nodes of Ranvier, one of the more poetic sounding of the structures in the brain. But the important thing to remember here is that the myelin is really a protective and insulating layer that prevents the dissipation of the action potential as it's propagated along the axon. Now here's something that's interesting. You've probably heard the brain referred to as your gray matter. And in fact, the brain is made of gray matter and white matter. The gray matter is the neuron bodies and the dendrites. So the gray matter is made of this part of the neuron. But the white matter, which sits sort of interior, the white matter is actually made of the myelin sheaths, because myelin has a whitish color to it. So if you look at a cross-section of a human brain, you'll see that the outside is much darker than the inside, and that's mostly because of the myelin that's insulating all the axons in the deeper tissue of the brain. So the action potential jumps from node of Ranvier to node of Ranvier here. in a process called saltatory conduction. Saltatory because we're talking about ions or salts. It's hard to visualize that, so let's take a look at a video summarizing the action potential.
This baseball player springs into action, his nervous system rapidly processing information in a network of nerve cells, or neurons. Each neuron receives input from one or more cells. In response, the neuron may generate an electrical signal known as an action potential that travels down the length of the axon. Let's take a closer look at the plasma membrane of an axon. Even without an action potential, the axon is a busy place with many ions moving across its membrane. Much of this ion movement is driven by the sodium-potassium pump. Using energy from ATP, sodium-potassium pumps actively transport sodium ions out of the cell and potassium ions in, creating an uneven distribution of charge across the membrane. Some potassium channels are open all the time, allowing potassium ions to leave the cell. As a result of these ion movements, the inside of the cell is negative relative to the outside. This condition is called the resting potential. The membrane of an axon is also packed with gated ion channels that open and close during an action potential. At resting potential, the gated channels are closed. If a stimulus changes the distribution of charge across the membrane sufficiently, the gated sodium channels open. Movement of sodium ions across the membrane makes the inside of the cell more positive. This reversal of the charge distribution causes the gated sodium channels to close and the gated potassium channels to open. As potassium ions move out of the cell, the original charge difference is re-established across the membrane, closing the gated potassium channels. This sequence of events is called an action potential. The sodium-potassium pump restores the distribution of ions back to their previous levels at resting potential. Let's look at how the action potential is conducted along the axon. As the change in charge difference across the membrane spreads from open sodium channels, other sodium channels farther along the axon begin to open. The original sodium channels close, and adjacent potassium channels open. As potassium ions move out of the cell, the original charge difference across the membrane is restored, and then the potassium channels close. Meanwhile, new sodium channels open, followed by the opening of new potassium channels and the closing of sodium channels. In this manner, the action potential is propagated along the axon of the neuron, eventually reaching another cell. The information carried by this action potential will be processed with other information, permitting this baseball player to make a spectacular catch.
So now that we've seen how a neuron can be innervated by a presynaptic neuron and can transmit an action potential down its own axon, let's take a look at what happens to that space in between the two neurons. So where one presynaptic neuron has been carrying an action potential, how does it then cause a change in the postsynaptic neuron? To understand that, we have to take a closer look at the structure of the synapse. So this is a great picture here. This shows a postsynaptic neuron cell body, and you can actually see these terminals where another neuron, or many other neurons, are trying to send signals to the postsynaptic neuron, and you can see how dense the connections are. It's interesting that the connections are actually indicators of learning. Every time we learn new things, we make new connections between neurons. And so intelligence is not really measured by the size of the brain or the weight of the brain or anything like that. It's really just a matter of how many connections you have, how rich are the connections between your neurons. Every time you learn something new, you're making new connections. And every time you review that thing that you learned, you're reinforcing those connections and making them stronger. That's why your teachers always tell you that you should reread your notes as soon as you take them, or study the night after you learn something rather than the night before a test. We tell you that. We hope it's effective. But anyway, we're going to look at chemical synapses, the method that one cell can use to communicate with another cell that it's not actually touching. Because most synapses are chemical synapses. Most synapses use chemical messengers to transmit messages from one neuron to the next. Not all. There are a few neurons that use what are called electrical synapses, and they use current from one neuron to send to another, and that's sent via a gap junction. Remember, a continuous cytoplasm between the two cells. Just a side note here, when I was at UCLA, one of my favorite places to eat was called Cafe Synapse, because it was the cafe in the Neuroscience Research Institute. So it's a great place to have lunch. It's actually within walking distance of Marymount, if you're ever interested. That was my college hangout. So here we have a schematized drawing of a chemical synapse. So we have an action potential traveling down an axon and eventually arriving at the presynaptic terminal. So this is the presynaptic neuron's axon right here. This is its terminal, or its ending point, on its axon. And so the action potential has been depolarizing until finally it depolarizes the end of the axon. In response to that action potential reaching the end of the cell, the depolarization triggers calcium channels inside the presynaptic neuron to open up. And as a result, calcium ions rush into the cell. So we have calcium ions rushing into the presynaptic neuron. Now you might ask, well, we're trying to cause an effect in the postsynaptic neuron. Why are we bothering with the presynaptic? Well, that's because the electrical change happens in the presynaptic. But what we're going to do is cause a chemical change here to affect the postsynaptic. So the opening of calcium ion channels allows calcium to rush into the presynaptic neuron, and the release or the influx of calcium triggers the synthesis of these vesicles, and the synaptic vesicles then fuse to the cell membrane and release tiny bunches of neurotransmitters into the synaptic cleft. So these little red dots here indicate neurotransmitter molecules, and you can see that they're being released by exocytosis from the presynaptic neuron. Those neurotransmitters are what cross the synaptic cleft and, with any luck, bind to some postsynaptic receptors on the postsynaptic membrane. So here's our postsynaptic membrane. We have lots and lots of receptors here, and here's a close-up of just one. So on this postsynaptic membrane receptor, we've received and accepted a neurotransmitter molecule. That neurotransmitter molecule triggers a change in the shape of that receptor, and it now opens up to either sodium or potassium, depending on what kind of receptor we've activated. As a result, we now trigger an action potential in the postsynaptic neuron. Now, one really big issue is how to take care of this neurotransmitter once it's been released. We usually release a lot more neurotransmitter into the synaptic cleft than we need to. And so the neurotransmitter is usually, it can be taken up by the presynaptic cell, so it can try to reattract those neurotransmitter molecules, or the neurotransmitter molecules are simply allowed to diffuse out of the cleft, and then they'll be taken up by surrounding cells and then get degraded. So we don't want too much neurotransmitter released. Otherwise, what you risk is desensitizing the postsynaptic membrane because of constant activation by the neurotransmitter. I mentioned before that when the neurotransmitter binds to the postsynaptic receptor, it can either trigger the opening of sodium gates or the opening of potassium gates. And it's important to consider which it is. If sodium rushes into the postsynaptic cell, then you trigger an action potential. Because remember, action potentials started when sodium ions open up and sodium rushes into the cell. That's called an excitatory postsynaptic potential because we're triggering an action potential. This is EPSP for short. On the other hand, if you're triggering the opening of a potassium ion, or potassium gate, then you're actually reestablishing or strengthening the membrane potential because what you're allowing is for potassium to rush out of the cell, and the cell becomes even more polarized. It becomes even more negatively charged with respect to the outside. So that's called an IPSP. It's sort of like the opposite of an action potential, and it makes an action potential even less likely to occur than it would have otherwise. You've probably heard of a lot of neurotransmitters before. One really great one is acetylcholine. It's incredibly active in our bodies. It is the neurotransmitter that works on muscles. And we'll talk more about muscles in our next class. But acetylcholine is the neurotransmitter that is released by neurons that activate your muscle cells. So every time you move, you're using acetylcholine to activate your muscles. And a little bit of background on acetylcholine. If you've ever heard of Botox, it's the treatment of wrinkles. And what Botox really is it's a form of botulinum toxin. It's actually the same kind of bacteria that grow in canned food that hasn't been properly sealed. And it can be deadly if it's ingested, but a much more mild form of botulinum toxin is injected, and it's called Botox for short, but really it's a toxin. And what it does is it inhibits the release of acetylcholine by your nerve cells. What that means is that you then can't activate the muscles in your face, and so you can't squint and reinforce those muscles, those wrinkles that form around your eyes. You can't furrow your forehead. You can't, make certain facial expressions for a while, and that's supposed to ease the wrinkles that form naturally in your face. I'm not taking a stance for or against Botox treatments, but take what I've said to heart. That's what you're doing, injecting botulinum toxin into your face in order to block the presynaptic release of a perfectly naturally occurring neurotransmitter, and in the process preventing yourself from making certain facial expressions. A couple of really important neurotransmitters in terms of mood regulation are serotonin and dopamine. And finally, endorphins are neurotransmitters that reduce your pain perception. So the LA Marathon is coming up, and those runners, once they hit a critical point in their running, will stop feeling the pain in their joints and their muscles from all of this intense exercise, and the endorphins will be released, and they'll allow the runners to keep on running. It's called runner's high once that pain is converted or ceases to be felt because of the action of endorphins.